Bio Radio.
So today on BioRadio, we will be discussing the effects of tree tapping and why it doesn't result in a tree being permanently damaged. We will be looking at how the tree can heal itself and recently discovered processes that prevent the tree from bleeding to death. When we say bleeding to death, we are referring to sap continuously flowing out of the tapped part of the tree. Today, we are bringing in a couple researchers to explore this topic. Hey guys, thanks for joining us. So tell me, what studies have you done recently in this field, and how are they different than what is already known?
Previously, methods were very complicated in order to study wood at the nano level. But now, thanks to the laser scanning microscope called the 4PI, scientists have discovered that the pit system within trees are composed of a membrane of nanofibrils that resemble the bullseye of a target.
Now, I'm not a tree expert, but what exactly is involved in this pit system?
Due to this new technology, scientists are now aware that when a tree is injured, torus, or the bullseye part of the tree, and part of the membrane shift from center of the pit to one side to seal the pit opening. This helps explain how trees seal off the cells of wood so that softwood trees don't bleed to death or lose all their sap when they are injured. Without pectin, a tree can't defend itself and it can't seal off damaged cells.
So what is pectin?
Pectin is a carbohydrate that is contained in the cell walls of plants that acts as a sealant when a plant is injured.
So what happens when a plant is injured? Kelly, you're an expert in this area. Can you explain to the listeners what mechanisms are responsible for plant defense?
Well, to prevent injury, plants have pre-existing barriers for defense, such as the cuticle and the bark, that shield the inner plant tissue. Thorns and trichomes keep herbivores and insects at bay. If a plant does become wounded, the stem tissue becomes disconnected and new tissues must be generated to restore connectivity. Because plant cells aren't mobile, each cell must be capable of defense. This defense is dependent on the transcription of certain genes that lead to the production of signals for healing and defense mechanisms. The signals trigger metabolic responses locally and systemically, but only the locally damaged tissues undergo a severe disorder of cellular structures that leads to water and nutrient loss.
In the next few minutes, we will be exploring how trees manage damage in 3 different ways: through phloem regeneration, bordered pits, and pectin. Then we will learn how a tree signals it has been injured throughout the tree itself. So Kelly, how long does it take for trees to recover from being injured or tapped? Is it like minutes or days?
Now, that's a very interesting question. What we know is that phelogen is responsible for secondary growth, giving rise to the periderm. Researchers studied phelogen's role in plant cell regeneration by drilling 2-millimeter holes into peach bark that injured the phloem, phelogen, pheloderm, and cortex of the tree. They took samples of vascular tissues in these injured areas after 3, 7, 10, 14, and 17 days. They then used special techniques to observe suberization and lignification by looking at autofluorescence with a microscope photometer. And here's what the study revealed. The white shapes on this graph indicate the appearance of new phloem in the tree tissue. The new phloem was observed 7 to 10 days post-wounding, reaching its maximum thickness at 10 to 14 days and then decreasing. New suberin, indicated by the black shapes, appeared 10 to 14 days post-wounding, and its thickness increased steadily with time. These results indicated a 2-step recovery process for damaged cell tissues. In the first phase, the cell wall was modified by lignification and suberization of tissues that were present at the time of wounding. The second phase included the generation of new phelogen and its derivatives. The initial lignosuberized zone was formed from the wounded cells after about 7 days, and the new phelogen formed interior to this zone 3 days later. The new phelogen pushes outward, crushing the lignosuberized zone. This process reestablishes the continuity of suberin and phelogen within the vascular tissue after wounding, and the damage is repaired.
Molly, you can speak to this. If plants cannot think or remember, how does a plant even know that it is injured?
Wound signaling is a very complex and interesting process in plants. There are many different organs involved due to the long-distance signals that must be sent. The higher the plant, the longer the signal it must send. This includes hydraulic signals, chemical signals, and electrical signals. Although the central role for jasmonic acid in plant responses to wounding is very well established, other compounds, including the oligopeptide cysteamine, oligosaccharides, and other phytohormones such as abscisic acid and ethylene, as well as physical factors such as hydraulic pressure and electrical pulses, have also been proposed to play a role in wound signaling, but many of the specific roles that they play are still not known. So, the most important hormone used in wound signaling is jasmonic acid, which is a plant hormone molecule that is produced in the wounded tissue and acts as a long-distance signal. When an insect attacks a plant, an enzyme triggers a response by jasmonic acid. The jasmonic acid activates the expression of protease inhibitors along with other anti-herbivore defense compounds. These protease inhibitors prevent activity of the insect's salivary proteins, which stop them from acquiring the needed nitrogen for the protein of their own growth. Therefore, if the salivary proteins do not have the nitrogen needed for the growth of their protein, then the salivary proteins cannot destroy the plants. In addition, another flow-mobile signaling hormone, auxin, plays a role in the transmission of the message that the plant has been wounded. In a recent study done, it was seen in wounded tobacco plants that when the apical meristems were removed, which is where the auxin exists, the plant produced less tobacco and took longer to recover. Auxin serves as the plant's neurotransmitter and therefore inhibits many wound-induced responses that occur when a plant is injured. Another chemical that may be used in wound signaling is hydrogen peroxide. It has been seen that after injury, a plant maximizes its production of hydrogen peroxide about 4 to 6 times what it usually is. Therefore, although the exact use is not known, hydrogen peroxide is definitely used for signaling the plant that it has been injured. So, there really are a lot of different chemicals and hormones that work together to signal the plant that it has been injured and responses to help heal it.
Thank you, Malvi, for going into specific detail on a complex subject for our listeners. Now, Keely, one of the things that you said you were able to observe with the new microscope were bordered pits. Would you explain to the listeners what bordered pits are?
Yes, they are especially common in the tracheids of some gymnosperms. Their structure is seen best in a cross-section through neighboring cells, which is where the microscope came in handy. So the highest resistance to sap movement is at the connections between xylem elements where sap moves through very much smaller holes in the cell walls between the ends of the cells. These small holes are the bordered pits. The interaction between sap and the walls of the xylem tubes create friction when the sap moves through the cells. The bordered part of the pit just means that there is an overhang that keeps the center called the torus, which is made up by a disc of primary cell wall material. The pit membrane, which lies in the center of each pit, allows water to pass between xylem conduits but limits the spread of embolism and vascular pathogens in the xylem. In addition, when a plant is injured, the carbohydrate, pectin, which we mentioned previously, forms ring-like structures at the margin of the torus that seals the pit and blocks the aperture.
We've been through a lot today. Jacqueline, can you wrap up what we've learned?
In conclusion, plants use phloem regeneration, bordered pits, and pectin to prevent from further injury when damaged. First, plants protect an injury initially using suberin and then phloem after 14 days, which increases thickness over time. Next, bordered pits aid to the recovery of a plant by allowing for the primary cell wall to stabilize through the overhang that maintains the torus. Lastly, pectin aids in sealing off the torus to the outside to prevent sap loss.
Well, that's all the time we have today. Thank you guys for joining us, and thanks to all our listeners out there. Learn more on our website at www.bioradio.com. Coming at you tomorrow is how to distinguish dinosaur fossils from aged rocks. I'm Lauren Wolcott with Bio Radio, and
We love botany!
Bio Radio.